As you read through manuscripts, do you find yourself saying, "The figures are difficult to interpret," or "The data do not seem as strong as the hypothesis suggests," or "I wish my results looked like that"? How you present data in your manuscript matters. It influences the perception of the entire work. That influence is very powerful. While authors want to present their best work in manuscripts, they must be careful not to make their results seem more impressive than they actually are. To obtain impressive results requires a solid hypothesis and a well-designed experiment. It requires good experimental technique and thoughtful data analysis. To acquire publication-ready data, you also must be willing to accept criticism from your peers and experts, and revise the study accordingly. Good research and good results take time. We, as researchers, should not create the illusion of good data by manipulating poor results. As noted in the American Chemical Society's guidelines on the ethical practice of research, an author's central obligation is to present an accurate and complete account of the research performed, absolutely avoiding deception, including the data collected or used, as well as an objective discussion of the significance of the research. Data are defined as information collected or used in generating research conclusions. These are very serious words. While the vast majority of researchers have no intention to deceive the public, some data preparation practices lend themselves to data enhancement. We all have to be careful about how we prepare our results for presentation in publications, as well as in grant applications, at meetings, and any other public venue. While research expertise varies, I bet our general methods for generating data are similar. What are some things you do before starting an experiment? First, I review the literature, develop a hypothesis, establish or determine a protocol, determine sample size, and define a method of analysis. What about during an experiment? Some ideas are validate the reagents, keep detailed notes, follow the protocol, and avoid distractions. Finally, what about after an experiment? We would analyze results, troubleshoot problems, repeat experiments, and discuss findings with colleagues. Are any of your labs the experts in a certain technique? The researchers ask your lab for help with the technique. What did they ask for? Why does your expertise matter? You know your criteria for good results in your work, but how do you assess whether results reported in a manuscript are solid? One can review the results, read the legends, read the methods, and consider whether the interpretation of the data correlates with the data that is presented. Are there other ways to assess the validity of published data? You might email the authors, call the lab, share the work with colleagues, or discuss the paper in journal clubs. When you are reading an article that is in your field, you are likely to be much more critical of the reported findings because you know what type of information the author should share with the reader, right? But what happens when the field is not in your area of expertise? What should you do to help you determine whether the study was performed well? The following are a few examples of how one could begin to determine whether the data presented tells you what you need to know to interpret the results. First, I am going to show you 3 studies that report similar results. Just to familiarize yourself with the experiments, each study is assessing whether animals change body weight due to muscle loss after having their hind limbs suspended for a period of time. 2 of the studies use myostatin knockout mice. I only mention this detail because animals lacking myostatin protein expression grow huge muscles, and the mutation is found in cows, humans, and other animals. On the right is an example of a myostatin knockout mouse that can be compared to a normal mouse. Here is a picture of a myostatin knockout cow. The first example to be considered compares the body weight, on the y-axis, of control mice and myostatin knockout mice with and without their hind limbs being suspended, as indicated by HS and C respectively. The legend for the figure is also provided. So, what information does this data share that is useful to the reader? Well, to me, I can see good aspects to this presentation, as well as things that might be lacking. For example, body mass is clearly displayed. The statistics are clearly noted in the legend, and the experiment is well described. However, there is no indication of how many animals were evaluated. Without that information, it is hard to know whether the results are derived from an appropriate number of animals, right? Would you believe the data if the results were from just 1 or 2 animals per group? What about 6 or more? Since the information is not in the figure or legend, where might that information be? Perhaps it is in the methods section or in the results section of the paper. Would it be more useful to have it noted in the figure or legend? The next example uses control wild-type mice and myostatin knockout mice to assess changes in body weight during and then after tail suspension. As before, there are some good aspects to the presentation, as well as some things that are lacking. Good aspects include an x-axis that describes experimental variables, and a figure legend that describes the experimental design and includes the number of animals analyzed. The changes in weight over time are clear and easy to see. However, we aren't told the absolute body weights for the animals. The use of relative changes in weight may mask important data. Notice also that the scale is truncated at 94, making the changes appear more impressive than they really are. This last example uses rats rather than mice as the experimental animal. One set of rats was free to walk in a cage. The other set of rats had their hind limbs suspended. In each set, body mass was measured at the beginning of the experiment and after 7 and 14 days. Notice that the table is descriptive, that a large number of animals were used, and that the statistics are included explicitly. However, because the authors have chosen a table to depict the data, it is less easy to visualize the differences between the two experimental groups. Although these 3 examples are similar, each shared its data with the reader in a different way. Each included some good aspects of presentation, as well as some ambiguities. It is worth taking the time to consider how you want to share your results. What information is important to your reader? What information is not? Remember that you will want to use the legend to provide details that a reader would need to assess the results. Data can also be captured digitally via photographs of results in real time. Often these provide stunning images that help to visualize the reported results. Digital images can be presented in many ways, but there are a few elements that can help to make their interpretation easier. Let's look at a few examples. These are photographs of carotid arteries from wild-type control mice in the top row and from mice with a mutation on the bottom row. The photographs were taken of samples at the beginning of the experiment and 1 week and 3 weeks post-injury. Are there things that the author has done well in presenting these findings? Well, we can easily visualize the histological changes over time in the 2 types of mice. We are told how many animals were in each group, and each image includes a scale bar indicating 100 micrometers. Because all the photos are depicted to the same scale, they are easily compared. Moreover, the authors have used arrows to direct the reader's attention to the relevant results in the mutant animals. However, there are no arrows in the control animals to which the results are to be compared. Are you told that these are representative images of the results? Let's look at another histology example. Here are stained images of kidney tissues. As you can see, the legend is quite detailed. The authors have provided two sets of images. Notice that the top set, A, is at 200x magnification, and the bottom set, B, is at 400x. What have the authors done to help present the data clearly? As mentioned, the figure legend provides a detailed description of the experiment. Two magnifications are provided, and arrows are used to direct the reader's attention to the relevant results. However, we are not told how many samples were evaluated, nor are we provided with scale bars. How about the results from immunoblotting? In this image, we are looking at protein changes in samples of fetal and adult sheep carotid arteries. What are some of the ways by which the authors have presented these findings well? We are shown a large portion of the image that includes standards, in this case a set of protein controls, which allow us to compare expression levels. Moreover, the figure legend is very detailed and describes the analysis. Indeed, perhaps there is a bit too much detail in the legend, and some could be reported in the paper itself. Notice that the relative expression is also reported in a bar graph, allowing both visual assessment of the blots themselves and an easily interpreted depiction of the data. In this last example, we are looking at protein expression in samples from the kidneys of control and experimentally treated rats. The authors have presented the relative expression of a few proteins, in this case, COX-2, NOS1, and NOS3. How have the authors presented these data well? Notice that there is an image to go with the relative expression depicted in the bar graph. However, we aren't told how many samples were used. The controls are standardized to an expression of 1, but note that 1 is not the same for each protein. Some of the proteins are poorly expressed, and some are highly expressed. More troubling is that the image is in pieces. Are they all from one experiment? Are they really comparable? So, these examples illustrate just some of the things to consider when deciding how much information you are being provided and what information may be missing. These issues become important because there have been a number of studies stating that a lot of biomedical research is not reproducible. That is, when other researchers try to repeat published work, they can't. Why? Some think that the studies are done poorly, that the details about the study are lacking, or that the reported results are not interpreted accurately. One researcher, C. Glenn Begley, has reviewed the preclinical cancer literature to assess why studies using animals to identify cancer treatments rarely translate into therapies for humans. He identified 6 red flags for identifying work that will likely not be reproducible, and he came up with 6 questions a reader should consider when evaluating a research paper. Were experiments performed blinded? Were basic experiments repeated? Were all the results presented? Were there positive and negative controls? Were the reagents validated? And were the statistical tests appropriate? These are great questions to consider, not only for the preclinical cancer research field, but for almost any other study. It is good practice for you to consider whether you yourself are running well-designed experiments and can report the findings with enough information for someone else to repeat the work. Although performing and presenting good experiments is important, it is absolutely critical that you have the original data available for you, your co-authors, colleagues, or journal editors to assess. Yes, even journals may ask you to upload data as part of the submission process, or they may ask to see the raw data during review. Thus, you have to have a data storage system. This system should be one that can be interpreted by your advisor, a lab mate, or someone unfamiliar with your work. Think about it. Would your advisor be able to locate readily a piece of your data if you had to instruct him or her over the phone or by email? If not, now is the time to set up a record-keeping process. At a minimum, you will want to label your data with the date of the experiment, the lanes or samples tested, and the treatments or variables given. You will then want to save all versions of your data set. If you modify the data, save the original and the modified version. Finally, make a special note of data that is used in a manuscript or public presentation. These pieces of data should be easily located and saved in multiple places. Grant funders like the NSF and NIH require grantees to save all components related to a grant for at least 3 years. They define the 3 years as starting after the final report is submitted. Since you may not know when this occurs, I would save the raw data and any data used in publications for even longer. 5 to 6 years isn't out of the ordinary. This is likely longer than you will remain in the lab. All your papers, notebooks, and computer files should be saved. Computer files should be saved on your institution's servers as well as external hard drives. Computers break, floods happen, files disappear, and people leave the lab. For these reasons, save your data in multiple places. Excuses do not replace lost data. Keep in mind that the data are owned by the institution. They do not go with you when you leave the institution. You may make copies, but the originals should stay. Once your studies are published, the obligation to have access to your raw data doesn't go away. Readers, your institution, and even scholarly journals may ask to see your raw data. You should be able to provide it, at least for 5 to 6 years after publication. This video provides an example of why data management is important. Now, let's complete the online exercises.